The next mechanism of ALS that I will discuss is the neuroinflammation hypothesis. However, to fully understand the role of neuroinflammation in ALS, we have to make sure you know what the immune system is and its different components. The immune system generally refers to a diffuse collection of cells and organs that are responsible for the ability to resist infection and disease. Immune responses will depend on the activation of white blood cells. White blood cells are born in the red bone marrow and can give rise to two different lineages of cells, the lymphoid lineage or the myeloid lineage. The lymphoid lineage will give rise to lymphocytes, which includes T cells, B cells, and natural killer cells. The myeloid lineage will give rise to many cells, one of which is monocytes, which eventually becomes macrophages. In addition, the central nervous system contains glial cells, which are the cells that support and protect nerve cells in the brain and spinal cord. While not all glial cells are considered immune cells, microglia, a specific type of glial cell, are considered the immune cells of the brain. In addition, astrocytes, another kind of glial cell, is a crucial regulator of the immune responses in the central nervous system. Lastly, the immune system also comprises of the complement system, which involves complement proteins that, when activated, will generate an enzyme cascade, eventually leading to an inflammatory response. Now, neuroinflammation in ALS is defined by the activation of reactive microglial and astroglial cells, as well as the appearance of lymphocytes and macrophages and the involvement of the complement system. I'll discuss a few studies on each of these and show how they might be related to ALS. Microglia activation has been found in the brain of patients living with ALS and in mice with a mutated superoxide dismutase 1 protein, or SOD1. As you might recall from the oxidative stress section, SOD1 is an enzyme which plays a role in breaking down toxic molecules. Importantly, these patients were shown to have a significant correlation between the intensity of microglial activation in their motor cortex, a part of the brain, and the severity of their motor neuron deficiency, meaning the greater activation of microglia, the more damage there was to motor neurons. Now, the mechanism behind this is as follows. Motor neurons and astrocytes that are damaged or are undergoing degeneration due to ALS will release misfolded proteins, such as mutated SOD1 proteins, which will activate microglia by binding to their surface receptors, such as the CD14 receptor and the TLR2 and TLL4 receptor. Now, to prove that motor neuron degeneration was being caused by activated microglia, researchers replaced the microglia producing mutated SOD1 protein with healthy microglia, as well as microglia that had a reduced expression of mutated SOD1 protein. The results show that this transplant not only delayed motor neuron degeneration, but it also extended the lifespan of these mice. There have been many in vitro and in vivo studies performed that have shown that astrocytes expressing mutated SOD1 protein have been shown to be toxic to both healthy motor neurons and to motor neurons of mice carrying the mutated SOD1 gene. While in vivo refers to experiments conducted inside a living organism like a mouse, in vitro refers to experiments performed outside a living organism like in a petri dish. Researchers have been able to find a way to selectively block the expression of the mutated SOD1 gene in astrocytes, resulting in a reduction of motor neuron toxicity and motor neuron death caused by the astrocytes. Moreover, the blocking of mutated SOD1 in astrocytes caused a delayed progression of ALS and lengthened the lifespan of mutated SOD1 mice. These same results were replicated when researchers transplanted healthy astrocytes into mice with mutated SOD1 genes, rather than selectively blocking mutated SOD1 expression. Then, when researchers transplanted astrocytes expressing the mutated SOD1 into healthy rats, it had the opposite effect. Rather than reducing motor neuron toxicity and slowing disease progression, it actively triggered motor neuron degeneration and neuron death. As previously mentioned, T lymphocytes are a type of white blood cells, and their job is to detect and respond to foreign or cancerous substances. Specifically, CD4 T cells, also known as helper T cells, recognize antigens and release cytokines to activate other immune cells. Cytokines are just small proteins important for cell signaling in the immune system. CD8 T cells, also known as killer cells, directly attack and kill the invader. Researchers observed that in the spinal cord of mutated SOD1 mice, there were normal levels of CD4 T cells at the early stages of the disease and normal levels of both CD4 and CD8 T cells at the end stage, which is the later, more severe phase of the disease. However, this trend changes for regulatory T cells, which are involved in immune suppression. Researchers found that there was an increased number of regulatory T cells in the spinal cords of mutated SOD1 mice at the early progressive disease stage, along with an increased expression of neuroprotective microglia. Conversely, in the later phase, there was a reduction of regulatory T cells cells accompanied with the loss of FOXP3 expression, which is the transcription factor for regulatory T cells. In other words, it's the protein that allows FOXP3 to be expressed, which therefore allows regulatory T cells to be made. Now, recognizing this, these researchers conducted a transplant experiment where they directly transferred CD4 T lymphocytes from a healthy mouse into an ALS mouse which lacked functional T lymphocytes. The results showed that this lengthened disease duration and prolonged survival of the ALS mice. Another study conducted by different researchers found a similar result. After removing CD4 T cells, or both CD4 and CD8 T cells, ALS progressed rapidly and increased expression of NOX2, which is an enzyme that produces reactive oxygen species and pro-inflammatory cytokines in mutated SOD1 mice. Now, the reintroduction of both CD4 and CD8 T cells was found to prolong the survival of the mutated SOD1 mice and inhibited the activation of destructive microglia. Many studies have documented the activation of monocytes in the blood of ALS patients, in addition to higher levels of invasion of peripheral monocytes into the spinal cord of ALS patients and mice, which ultimately led to a greater motor neuron degeneration. Other studies have mentioned that activated monocytes in ALS patients showed reduced phagocytosis, which is a process by which certain cells remove harmful particles and dysfunctional pro-inflammatory secretion of cytokines. Researchers have found that the complement proteins including C1q, C5a, and C5b9 are increased in plasma, cerebrospinal fluid, and the spinal cord of ALS patients and mutated SOD1 mice. Furthermore, after activating and increasing the expression of C5a, C5aR1 signaling, which is a component of the complement cascade and plays a crucial role in the immune response, led to an increased migration of macrophages, which was suggested to explain the observed accelerated muscle denervation and motor neuron death in mutated SOD1 mice. Lastly, after researchers selectively blocked the expression of the C5a, C5aR1 signaling, mutated SOD1 mice were shown to have reduced motor symptoms and an extended survival.